Okay, the last big topic of Lecture 2 is the cell structures. We're going to look at what makes up a bacterial cell. And we're going to start with what's in the cytoplasm, or inside the cell. And the first thing we're going to look at is something you already know. It is the ribosome. Bacterial ribosomes do the same thing human ribosomes do. They are giant complexes of protein and RNA, just like human ones, and they synthesize new peptides based on messenger RNA nucleotide sequence. They are structurally a little bit different because our common ancestor lived like 3 billion years ago, but they enzymatically do the same thing. Bacterial ribosomes are similar to other bacterial ribosomes. Archaeal ribosomes are like other archaeal ribosomes. Eukaryotic ribosomes are like other eukaryotic ribosomes. But within a domain, or between domains, they're very different. But they all do the same general function. Okay. What you should know, though, about bacterial ribosomes first is that they are very abundant in fast-growing bacteria. In bacteria that are growing exponentially with a doubling time of like 20 minutes, ribosomes make up by far the most protein in the cell because they are frantically making proteins so that they can frantically make new cells. They're just full of ribosomes, and also the ribosomal RNA is also very abundant, and it's much more abundant than any other larger RNAs like messenger RNA. So if you do something like you run the bacterial RNA out on a gel, you will see 2 bands for the 2 ribosomal RNAs, and you won't see anything else because those ribosomal RNAs are so much more abundant than all the messenger RNAs. And I don't think that might be the case in human cells, but it's definitely the case in bacterial cells. So ribosomal RNA, by far the most abundant. The ribosomal structure should look vaguely familiar to you, but these numbers would be different because they are different sizes than what you saw in humans. The S stands for, I believe, the Svedberg units. It's just like how fast it sediments in a centrifuge. It's not that important. The only one you need to know is the 16S ribosomal RNA. So this giant subunit that has 20 proteins and 1 RNA is what actually does the addition of the new amino acid. And the 16S ribosomal RNA, I believe, is the catalytic part that actually does that reaction, whereas all the other proteins and RNAs work together to position the peptides and the new amino acids correctly to hold the messenger RNA in the right place and stuff like that. So that ribosomal RNA ends up being really important, and it is the most fundamental part of the ribosome. Some people think it's the first part that ever existed, but it's definitely what we have used to construct the universal phylogeny. Humans have an analogous RNA. We don't call it the 16S. I believe it's the 18S. But if we trace it back to our common ancestor, it was the same thing way back when. So there's that. Okay, that's RNA. Otherwise, RNAs are the same. Sorry, we have the same, or bacteria have the same messenger RNAs and transfer RNAs. There are differences, but the same general idea in translation and protein synthesis in bacteria and humans. The DNA in a bacterial cell is called the chromosome. And one thing we have to think about is we have to distinguish a chromosome from some other DNA molecules you might find in a bacterial cell. So what's distinct about the chromosome tends to be that it's huge. It might be 5 million nucleotides long. And a cell needs the products encoded by those chromosomal genes always. So there are genes on the bacterial chromosome that are always necessary. They have to be transcribed all the time. And we're going to contrast that with what's called a plasmid, which is not always necessary. So the big DNA molecule inside a bacterial cell with the absolutely essential genes is called the chromosome. Bacteria don't have nuclei, so we don't find the chromosome in the nucleus. There is some spatial organization, though. Bacteria are not random on the inside. They're organized, they're just not, there aren't membrane-wrapped organelles. Remember, bacteria and mitochondria are related, and so bacteria are the size of a mitochondria. And picture a mitochondria and its relationship to any other part of a human cell. Bacteria are too small to have a lot of real organelles in them. It's not important. Okay, so the plasmid is the other DNA molecule I was talking about. Plasmids are shorter DNA molecules with not as many genes, and those genes are conditionally important. They're conditionally useful. So they're useful in some circumstances, but not in all circumstances. So they're not always required. So plasmids have these conditionally useful genes, but the real reason we care about them is that they can move between bacteria. Bacteria can pick up plasmids from the environment and say, oh, cool, this is a plasmid. What genes are on that plasmid? What abilities will this give me? And that is a big thing that happens in bacteria. They swap DNA. Antibiotic resistance is often encoded on plasmids. So if you've heard of KPC1 or NDM1, these are plasmids, well, no, those are enzymes, I should say, that inactivate some of our best antibiotics. So if a person gets infected by a bacterium that has one of those genes, we can't treat them with common antibiotics we're likely to have. So those genes are encoded on plasmids, which means that if one patient in your hospital has an infection with bacteria with that gene, it is going to interact with other bacteria and they will get that same gene because they will get that plasmid on. And so they'll keep replicating that plasmid DNA every time they divide, and all their daughter cells will have the enzyme too. So that's one reason why whenever you have badly antibiotic resistant bacterial infections, you have to isolate that patient because you don't want other bacteria getting the plasmid that has those genes. Also, virulence factors are often on plasmids. Virulence factors are anything that makes a bacterium able to cause a disease. So they could be little toxins they use to poison our cells, or really anything else that helps them with the disease. Okay, so that's what's in the cytoplasm. Next, we look at the cell surface. This should be vaguely familiar to you. This is a phospholipid bilayer, and in every textbook they draw it as a little sphere that represents the phosphate, and then the tails that represent the hydrocarbon chains. So you tell me, which one of those is hydrophilic? 3, 2, 1, pause. Whoops. Sorry. Yes, the heads, the phosphate heads are hydrophilic. The tails are hydrophobic. And that makes this phospholipid bilayer membrane have this hydrophobic core. And that makes it stable. It makes it hard for these lipids to flip or leave. And it also makes this waterproof, more or less. So you can get some water molecules moving through, but that's a slow process. So we use a phospholipid bilayer membrane to sort of designate the outside of the cell, and it separates the inside from the outside, and it's waterproof. Bacteria have an additional thing called a cell wall, and it's made of a polymer called peptidoglycan. And that polymer is made of sugars and amino acids. It's made of like sugar chains that are connected to each other using short chains of amino acids. This is important because I think more than half of the antibiotics that we prescribe, their function is to disrupt peptidoglycan. So the penicillins and the other beta-lactam antibiotics like the cephalosporins and the carbapenems and aztreonam, those all target the cell wall. They actually all work in an identical same way, disrupting the cell wall. Vancomycin works in a similar way. And again, I think, more than half of the antibiotics we prescribe do that. So that's important to know about. And so what happens when we disrupt the cell wall? Usually what happens is lysis. The cell lyses, and that is death because the cell contents leak out and the outside world leaks in. And the cell wall is going to be different between gram-positive bacteria and gram-negative bacteria. So I mentioned gram-positive, gram-negative in lecture 1. This is going to show you sort of what that difference is. This is showing you sort of a cross-section of gram-positive and gram-negative, positive, negative cell membranes and cell walls. And what this slide is showing you, just you can ignore this for now. What you should be thinking about is, why do bacteria need cell walls? And I'll tell you, they're not like shields. They're more like nets. They're more like a net. And they give the cell strength, while the membrane gives the cell waterproofness. And what this slide is telling you is that most bacterial cells have a lot of osmotic pressure inside them. And that's how they actually keep their shape. They're kind of inflated like balloons. So there's constant pressure pushing out on the cell, on the membrane, the bacterial membrane. If that happened to one of your cells, that cell would rupture. But a bacterial cell has to be able to handle that pressure. So that's why they have the waterproof membrane wrapped by the polymer, peptidoglycan. And if you stick around for the last video, I will both do some math and demonstrate how that works using a sock and a balloon. Okay, so enough of that. Let's think about peptidoglycan and lipids. So lipids are what make up the cell membrane. Peptidoglycan is strong. And we just think about lipids are weakly connected to each other, but they make a waterproof layer. You can dissolve them using detergents. None of that is true about peptidoglycan. Peptidoglycan is connected through covalent bonds, and it's physically strong. And to disrupt it, you have to use an enzyme or an extreme physical force. We can compare gram-positive to gram-negative cells, and we're going to see they do have a different structure. What they have in common is the waterproof cell membrane. But then if you look outside that, what you see is different. So if you look just outside a gram-positive cell membrane, you see a thick peptidoglycan cell wall. And that's what's in this slide. So it's a rod-shaped cell. And what do we call those? What's the cell morphology called? 3, 2, 1, pause. That's a bacillus. And what they're showing is if we cut a section of that cell wall out and magnified it, this is what we would see. This is the inside, this is the outside, this is the membrane with the phospholipid bilayer, and this is a thick layer of that peptidoglycan polymer. Just sheets and sheets of this tough polymer. And this is showing you the carbohydrate chain and the amino acids that connect it to the other carbohydrate chains. So that's what all this is. So waterproof cell membrane, outside of that is a thick peptidoglycan cell wall. Gram-negatives also have a waterproof cell membrane. Outside of that is a space full of water. It's called the periplasmic space. And they have a thin layer of peptidoglycan that is actually inside the periplasm, or inside the periplasmic space. The outer edge of the periplasmic space is defined by the gram-negative outer membrane. So if we check out a gram-negative, again, it's a bacillus, and we cut out a piece, and here's what we see. Inside, outside, cell membrane, this is waterproof, and then the periplasm is in between the cell membrane and the outer membrane. So this periplasm is all the stuff in between those two membranes. And there's a thin layer of peptidoglycan in that space. And that peptidoglycan has the same function as the thick layer in gram-positives. It prevents the cell from exploding. So if we go, well, let's continue. So this is a slide that talks about the cell membranes. They're similar to each other. They're waterproof. They allow the cell to decide what comes in and out. Like any charged molecule can't cross it, so they can choose which charged molecules go in using active transport. They can put channels in it to let passive processes work. The cell membrane, that inner membrane, that is where the electron transport chain is. That is where the proton motive force is created. So the protons are pumped across that inner membrane. So if you think back to what they call, I think, cellular respiration, which I will call aerobic respiration, in that process, something like sugar gets oxidized and the electrons that are taken off onto NADH are put into an electron transport chain, and they move through that chain to oxygen. And in the process, they pump protons to make a proton motive force. That happens at the inner membrane. So then gram-negatives are the ones that have a periplasm, and this talks about that. I just talked about it. But what the gram-negatives have that's more spectacular is that outer membrane. And it is a waterproof lipid bilayer, but the cell puts holes in it, small protein pores that let molecules in water cross it easily. So the cell doesn't use it as a waterproof barrier. The gram-negative outer membrane is not meant to prevent ions from coming in and out or anything like that. It's meant to prevent big things from crossing it, like proteins. And so there's no electron transport that happens at the outer membrane. There's no proton motive force. It's just kind of an outer shell. And if we go back to look at a membrane, well, let's look at a gram-negative. If we look at a membrane, it's a lipid bilayer. So there are two layers, right? They face each other. Each one has hydrophobic tails that face towards the inside. So we have this outer layer and this inner layer. They are best, I think, called leaflets. So this is the inner leaflet, and this is the outer leaflet. And if you notice, they are different. So there's all these dark colors out here, whereas this is all yellow. Well, this has some blue proteins that we're going to skip, but this has a lot of red proteins with green things sticking out of them, and we're going to talk about that next. So the outer membrane has an outer leaflet that has some unusual lipids. They're not all phospholipids. The ones that were red in that picture are called lipopolysaccharide, or LPS, or endotoxin. Those are all names for the same thing. LPS is only usually found in gram-negatives with a tiny number of exceptions, and we call it endotoxin. It has a lipid part and a carbohydrate part. The lipid part is embedded in the hydrophobic layer of the membrane. The carbohydrate part sticks out of the cell. It is easily recognized by the human immune system. So the human immune system notices it and says, oh, there must be gram-negative bacteria in here. And our immune system will quickly respond by triggering fever and inflammation. So we respond very strongly to LPS, and usually it's not released into our body until one of these cells dies and lyses. So that's why we call it an endotoxin, because it's an intrinsic part of the cell and they don't release it. It is inside or within that cell, sort of. We would contrast that with exotoxins, which are toxins that bacteria actively secrete and release. Endotoxin is an intrinsic part of the outer membrane. And so what that means is we can inject dead gram-negative bacteria into someone and cause a severe fever. You can kill someone by injecting dead gram-negative bacteria into them. And so any injectable liquid has to be tested for the presence of lipopolysaccharide. And if you look this up, you'll see that the most sensitive test for lipopolysaccharide is the blood of a horseshoe crab. I'm not going to talk more about that, but that's what they use. And so, yeah, if you look at like injectable water, it might say pyrogen free, and that means it doesn't have any detectable lipopolysaccharide. So again, there are two little groups of gram-positive bacteria, the actinobacteria and the firmicutes, two phyla. And there are lots of other ones that are gram-negative. And I definitely want you to know the structural differences between gram-positive and gram-negative, because we are going to look at how we control these cells, how we kill them, and how we prevent them from growing. And that really does depend on the chemistry and the physics of their cell structures. Okay, so I'll end this here, and we'll jump into the last video next.